So very good morning. Namaskar, everybody. So I'm very happy to be here. So thank you, Professor Gurnule, for inviting me. So thank you, everybody, for being here. In my lecture, I am going to talk about the topic that you can see on the slide. So I need a little bit time to be used to, it is okay, with this pointer. You can see that I'm going to talk about the inorganic nanoparticles coated natural fibers for two main applications. Okay, so I think there in this auditorium we have the scientists from different disciplines, perhaps physics, chemistry, mathematics, biology. So, but this topic is something more about biomedical application. So therefore, I am going to briefly discuss on this topic. So please don't expect that I am going to make a very big presentation. I am talking about very small things. Okay, only two applications. These are quite new works done in my group. Nothing is published. All is new. Hopefully good. Hopefully not good, we don't know, because we need more investigation. So let us go to the topic that I'm going to talk about. These are the topics. I want to start with the introduction. Then I will talk something on general methodology. Now let us proceed. We all know about the own and own healing is a complex process. And we know that nanoparticles, among others, are increasingly used in biomedical applications, also for wound healing and also for bone tissue engineering, the topics which we are talking today. So this is quite a complex process. So let me go to another topic about the bone, which is again a very complex material. Actually, a kind of nanocomposite material made up of some minerals, the hydroxyapatite, embedded in a very soft matrix of collagen. You can see in this picture I took from the literature that the bone has a hierarchical structure in different levels, from macro to nano scale. And this hierarchy of the structural details of the bone make it a kind of very unique material. A unique flexibility, not flexible like rubber, but it has some flexibility. Very strong material and very unique biological activity. So when we are talking about the bone tissue engineering, then we immediately come to the point that, okay, if there is a fracture in the bone, there is some loss in the bone, there is some damage, is it possible to repair it? It is possible, we all know that. And the process of inducing the bone regeneration or bone formation to bring it into new function, that we call tissue engineering, bone tissue engineering, and it has something to do with structure of the bone, functionality of the bone, the mechanical properties, mechanics of the bone, and the mechanism of the formation of the tissue. And there are several things utilized in the bone tissue engineering, but particularly many polymers, particularly the biopolymers, which are generally biodegradable. They should be like that. And the materials of the choice must possess properties like osteoconductivity, bioinertness. They should not be toxic, they should not be poisonous, and of course, they should be compatible to the biological tissues. Keeping these things in mind, let us come to very briefly the structure of the bone forming mineral, the hydroxyapatite. You can see in this picture the structure of the platelet-like structure of the bone, hydroxyapatite, the mineral that forms the bone. The crystal structure is shown here. This is a calcium phosphate-like material that is embedded within the matrix of very soft polymer, the protein, the collagen, and it has the hydroxyapatite that can also be synthesized in the laboratory, in chemical laboratory, has a major mineral component present in it is the major mineral component present in the tissues of the vertebrates and has high osteogenic potential. So now the motivation is not to discuss these things, but the motivation is if it is possible to utilize biopolymers, natural fibers, natural polymer, natural plant fibers in order to heal the bone and in order to encourage the bone tissue engineering. So can I have some water, please? So thank you. So this is a kind of old man problem, you know. When the old man speaks, he needs a lot of energy. Also, Professor N.B. Singh would say that I'm not old. So thank you. He smiles, he attests. So about the polymers, natural polymers I'm going to talk about. Using natural fibers and the paper thereof, because the paper is, I'm just talking about the paper, this kind of paper. It is also made from natural fiber. As a substrate for infection on bone healing, I'm sorry for the mistake, and for tissue engineering, bone tissue engineering application. So we know that recently, much research interest has been attracted by natural fibers and papers for promising scaffold materials for several tissue engineering applications. And the natural fibers that can be utilized to make papers like this, or different kind of papers. Of course, not very expensive, otherwise we would not use it so easily. They also can be made highly porous, no difficulties in oxygen diffusion processes. They are found to be biocompatible, bioinert, not poisonous, and can be made variably flexible. So, what about using the traditional Nepali paper for bone tissue engineering applications? This is also the question we put. So, the objective of this work has been, as I introduced, twofold. The first one is, of course, to extract the natural fibers from the plants and to coat them with some inorganic nanomaterials. In this case, we take zinc oxide and copper oxide nanoparticles. I have seen that in the lecture today and tomorrow, there are many papers related to copper oxide and zinc oxide nanoparticles for their biological activities, for their cosmetic applications, and also for their sensing application. I saw some papers. So our purpose is to extract the natural fibers, to coat them with these two kinds of nanoparticles, copper oxide and zinc oxide, and to see if they help in bone healing processes. This is the first. And the second is to prepare from those fibers the traditional way, the Nepali paper, we call it Nepali paper, it can also be Indian paper, and utilize this paper as a scaffold for attaching the hydroxyapatite nanoparticles that we can prepare in synthetic way, or we can also extract from biological tissues, and to see if there is a cell growth, easy cell growth, physical cell growth for osteoblast cells. So these are the main objectives. So how do we do that? For that, the general methodology is, first of all, as said, to prepare the lignocellulosic fibers, basically the cellulosic fibers, by removing the lignin, pectin, hemicellulose, and so on. And then to prepare chemical means by chemical means the nanomaterial and hydroxyapatite included, and to coat them, already mentioned, and perform biological tests for the potential application in respective fields. To make the elucidation of the structure, and of course, therefore, the properties of the materials that we have made, we utilize different techniques of spectroscopy, scattering techniques like X-ray diffraction, and electron microscopy. So in this picture, what you can see is, this is a representative of fibrous plant. Can be different kind of fibrous plant that we can utilize to make the papers, which is utilized to make the fibers, that is then converted to the pulp, and this pulp by some process can be changed into the papers, and the paper, if we have a look with microscope, for example, a scanning electron microscope, we see that there are the microfibers bound together, and this gives the paper the strength. So this is about the mathematical overview. So let us come to the results very briefly, without making very boring. So let me start with the preparation of zinc oxide nanoparticle. This is very easy method, can be found in all the literatures, or you can also discuss with the chemist in very easy way. So I don't want to discuss in detail, but I just want to tell you that, first of all, we have the zinc sulfate, we dissolve it in the water, we stir it, then we also add the powder of the natural fiber into it. For several hours, we can also make different kind of chemical treatment or physical treatments. Then we have the completely dispersed suspension of this substance into the water. Then after that, we add the caustic soda, and under given condition, we have the white precipitate, including that of the raw fiber, but this is of the hydroxide of zinc hydroxide. Zinc hydroxide upon centrifuging and dried at elevated temperature gives us a composite, nanocomposite of natural fiber with the zinc oxide nanoparticle. In the similar way, we can do copper oxide nanoparticle. The reaction is shown here in a very simple way. We do start with copper sulfate solution in the same procedure as before, and upon elevated temperature heating, it should be a little bit higher temperature, then we have black oxide of copper. This way, we can prepare copper oxide nanoparticles, embedded natural fibers. So now, for the own healing process. Already mentioned that copper oxide, zinc oxide, and many other kind of oxides and nanoparticles. Also, mineral kind of nanoparticles can be utilized for own healing applications. There are many works found in the literature. And nanoparticles do not have only these applications, they have many other applications. I'm not going to talk about. But how do we do this test for own healing? Own healing is also, as already mentioned, a very complex process. There can be the healing of the caught wound, it can be the healing of the infected wound, it can be the healing of chronic wounds that have several kinds of infection in series. For physicists and mathematicians, it is something difficult from. I just want to tell you, there is a process of making Mueller-Hilden agar medium into sterile petri dishes. Then we have made the gel, and we avoid any kind of contamination. Then finally, we have the samples. These samples are incubated, and in this Mueller-Hilden agar, after making some holes, and then we put our samples inside them and allow them to react, to respond by the microorganisms. The microorganisms are some so-called gram-negative and gram-positive bacteria. I'll come to that point. Before that, what we have is zinc oxide and copper oxide nanoparticles. They look like this. But this is not only the structure of zinc oxide and copper oxide nanoparticles. It depends upon under what temperature you create it. What are the processing conditions? What is the calcination temperature and so on? Our friend Raghuramji is here. He's an expert in the 3D5 mistake. Please correct. So, or I learned a lot of things from you, and if it's to try to make a mistake, it should be your mistake. Okay, under some conditions, we have the spherical zinc oxide nanoparticle. It can also be made rod-like or sheet-like or plate-like structure. And copper oxide nanoparticles, they have some grain-like, some rice grain-like structures. So they have different kind of shapes and sizes. Very quickly, without discussing much details, we can do the X-ray diffraction and the apparent diffraction pattern of zinc oxide nanoparticles and copper oxide nanoparticles are given here. So I don't discuss much on this. What we see is there are many peaks, for example, these peaks and these peaks here, which correspond to the Miller indices of the crystalline forms of these materials. So they attest that they are those particles of zinc oxide and copper oxide nanoparticles. More interesting will be when we look at the X-ray diffraction pattern of the composite material comprising the natural fibers and the nanoparticles. And that comes in the next slide. So if you just have a look before and after, we see that the nanoparticles, most of the peaks that were present before are also present here. That tells us that the nanofibers or the natural fibers have been completely coated, well coated with the nanoparticles. And if we have a look with scanning electron microscope in higher magnification, not very high magnification, but in a little higher magnification, we see that this is the natural fiber without coating. So you see that this is 100 micrometers. This is a bundle of 200 micrometers. One single fiber could be 10 micrometer or less. And all these bundles, but this is a small area. In another area, the structure could be similar, but bundles could be higher or smaller. So when we take this material, powder it, and then put with the zinc oxide nanoparticles, for example, then we see that these are raw fibers. This roughness onto the surface of the fibers comes from the deposition of zinc oxide nanoparticles. Okay, this is about the preparation and characterization of the fiber. So let us go to the result of this bone healing properties of this fiber. What we do, as introduced earlier, what we do is we measure the zone of inhibition dimension for common bone infection causing bacteria. And these are listed here. The number 2 and number 5 are so-called gram-positive bacteria, and others are the gram-negative ones. And for example, here we show the zone of inhibition produced by the reference material gentamicin. This is a common antibiotic that are used for combating the bacteria. And this is the fiber A, and this is fiber B produced in different ways. And this is for the zinc oxide nanoparticles. So this was for Bacillus subtilis, a gram-positive bacteria. Okay. So what we see is the zone of inhibition, for example, for gentamicin, this is measured by the diameter. Okay, and this is as good as the zinc oxide nanoparticle. And the fiber A and fiber B, which are coated with zinc oxide nanoparticles, are also not very bad in terms of the dimension of the zone of inhibition. So if we see the detailed result, what we see is this is for different bacteria. This is for this bacteria, another bacteria, and so on. What we see is the first line is for gentamicin, and the second one is for the zinc oxide nanoparticles, and the rest are for the fibers. Within the error range, we see that all the materials against the bacteria work quite well. The function is more or less the same. Also for another bacteria. So we see that for different types of treatment and different types of bacteria, the zinc oxide nanoparticles, the same is for copper oxide nanoparticles. The antimicrobial activity is quite good. And very similar to that of the common gentamicin. So this is for copper oxide nanoparticles. I don't want to go into detail. So what we find, to summarize, is successful coating of the natural fibers by zinc oxide and copper oxide nanoparticles were possible. And we found that under given conditions, the zinc oxide nanoparticles were spherical, while the copper oxide nanoparticles were rice grain-like, rod-like structures. And the nanoparticles coated fibers were found to possess quite good antimicrobial, antibacterial properties, both against gram-positive and gram-negative bacteria. And their performance was not very different than that of the common antibiotics, the gentamicin. So let me go to another topic very quickly. I think I already talked a lot. So how much time I have? Do I have, Professor? 10 minutes? 5 minutes? 10 minutes. 10 minutes is enough. So the second point is on bone tissue engineering. So on the picture you can see the scanning electron micrograph of, so this is acoustic microscopy image of the bone. You can see here the so-called Haber's canal, the so-called lamella, and all these structures, very organized structures. So I don't want to go to detail about the properties of this material, but I want to go for if we can do bone tissue engineering with natural fibers coated with hydroxyapatite nanoparticles. So hydroxyapatite nanoparticles can be synthesized by chemical means. Without going into detail, I just want to tell you that by adding under given conditions, defined conditions, the calcium chloride and disodium hydrogen phosphate solution, we can prepare the hydroxyapatite nanoparticles. And finally, it will look like this, the powder. 10 grams of hydroxyapatite nanoparticles may cost sometimes several $100, which we can, if we are good enough in chemistry, in synthesis, by utilizing biogenic sources or also by chemical means, we can prepare 10 grams of hydroxyapatite for maybe 200 rupees. So for this experiment, how we do, I just want to explain you a little bit about the experiment. What we do is, we take this paper. This is the paper under microscope, the traditional paper. The traditional paper, I mean the paper that our ancestors used to write the text, for example, Vedic text or Buddhist text, Pragya Paramita, something like that. They look like this under microscope. So what we do is, we take 2 centimeter by 2 centimeter pieces. I'm sorry, the picture is not so nice. Put that on the table, working table. Then we take calcium chloride and disodium hydrogen phosphate solution one by one. We take given amount of calcium chloride solution onto the paper. Then after that, onto the paper, you take the paper onto the paper. You put calcium chloride, and then after some time you put another solution. Okay. Then after that, the calcium phosphate like mineral hydroxyapatite is formed. So we do that in different cycles. We finish the cycle, we wait for another 12 hours, and we repeat the experiment. Then another 48 hours, 72 hours, and several days we can do. So finally, this paper turns to the paper like this. And this is that we are going to utilize as a scaffold for bone tissue engineering applications. So I'm not going to present you the real-time experiment with the bone. But what we are going to do is, we are going to take some cell line, which is representative of the bone cells. So this cell line, due to OS cell line, is tibia cell lines isolated from 15-year-old person. We did not extract ourselves. This is procured. We can buy it. And the subculture can be done. Cells develop an extracellular matrix, ECM, to adhere to the substrate. And that is called, the cells are trypsinized. So it is interacted with a substance called trypsin, that is called trypsinized, to separate the cells from the substrate. And with all this procedure, what we do is, after incubation for 24 hours, we get that ready for the experiment and then do MTT assay. So what is MTT assay for non-biological? What we do is, we have the subculture of the cell lines. So then we, after trypsinization, centrifugation, we have the cell pellets formation. Then we do it a resuspension of the cells in the medium. We count the cells, and we prepare the samples from the paper. And these papers, species of the paper, are put into the seeding cells, so-called 96 well plate. Okay. Then after that, we add into it the cells that we have prepared. Then allow to interact. By doing this, let me skip this. This is the structures. By doing this, we have, this is the traditional paper before scaffolding, deposition of the mineral, and this is after the deposition of the mineral. When we take the small pieces of the papers, then we see that in higher magnification, this is the lead paper, and this is after the mineralization. Okay. The mineralized paper, when we take it, after different mineralization cycle, we take the weight of the hydroxyapatite. We see that the amount of the minerals increases. Higher the number of the cycles, the higher the amount of the SAP mineral crystals as expected. So in the MTT assay, what I meant, it allows the estimation of the cell viability. There is a substance called MTT, the full name is given here. The chemical structure is given here. And when the cells are living, they interact with this substance, and it converts, the cells can convert the MTT into a reagent, that is colored one, that is called parmesan crystal. So by measuring, therefore, the concentration of the parmesan, we can determine, because this is if the number of the cells are higher, then more parmesan will be produced. By measuring the concentration of the Parmesan, therefore, we can estimate the number of the cells, living cells present. So therefore, which we can do by spectrophotometry. The concentration of Parmesan is proportional to the number of the cells. That can be done by spectrophotometry, and the result is shown here. So this is the cell viability as a function of mineralization cycle. Number of the cycles, that is the deposition of the hydroxyapatite more and more, then the more and more cells will be living there. So it means higher the number of the cycles, the higher will be the cell viability. The high concentration of HAP favors the cell viability. Cell viability for control was recorded to be 40%. So this result tells us not that about the structure of the bone tissues being formed, because this is not a real-time experiment, but it tells us that while utilizing the bone tissues which were procured, and when we are utilizing as a substrate the paper scaffold coated with hydroxyapatite, then it provides a ground to make the cells living. So it means by utilizing the paper scaffold containing the hydroxyapatite nanoparticles, there is a big opportunity, big chance of the cells to survive there, to proliferate there in the tissue. So the real-time experiment should be still done. That is in progress. The application of traditional Nepali paper seems highly feasible for its application as a scaffold with nanohydroxyapatite for bone tissue engineering. The research is going on. The next step is to do with real-time experiment utilizing the bone. This is we are working. So this is actually the end of my presentation. So I would like to thank all our collaborators from different countries, from Czech Republic, from Germany, and our undergraduate students who did a lot of work on it, Umisa, Manoj, and company, Sasikala, Shusmita, and Savita, and all the undergraduate students. Thank you for your attention. And I also would like to thank Professor Gohale and all the friends who were there to welcome us. Your welcome hospitality is really awesome. Thank you so much for your attention. I'm sorry if I could not make you understand everything very, make the things, everything very clear. But as one of the previous speakers told before, there is a need of collaboration among the countries, among the institutions, among the persons, among the scientists, and that I would also welcome very much. Besides this new area of biomedical materials, we are also work on polymer nanocomposites, polymer microcomposites, and as also mentioned by one of the speakers this morning, on microplastics and bioplastics. So very welcome, and thank you so much for inviting me again and for listening to me. Thank you. If there is any question, I will be very happy to.
Thank you for your nice presentation. And zinc oxide coated particles and copper oxide coated particles have compared to this. I mean, this antibacterial activity. And the zinc oxide coated particles, is less efficient than zinc oxide itself. I mean, zinc oxide coated fibers are found to be less efficient than the zinc oxide itself. I mean, that is it.
Yeah. In some cases, you're right. In some cases, the nanoparticles coated fibers are less efficient than the nanoparticles themselves. But it gives us the opportunity to reduce the amount of the nanoparticles. Yeah, which is also in turn sometimes not really undangerous. Okay.
Copper oxide is found to be more efficient. Copper oxide particles are found to be more efficient for the fibers.
Yeah. Which particles?
Copper oxide. Copper oxide.
Yeah. Why copper oxide are more?
They are found to be more efficient.
Yeah. They are more efficient. Your question is why? The answer is I don't
So
know.
the HEP, fibers,
Yeah.
in that you can also coat silver and other metal ion so that it will be more efficient.
Yeah. Yes. Yeah. You're right.
Thank you.
Yeah. Yeah. But to add to your question, we are still working on it, but it of course depends upon how exfoliated the fibers are, how big surface area they provide for the coating, and how the dimensionality of the nanoparticles is. So that are also very, very important factors. But you are right. Thank you.
So can I ask you one question?
Please ask me two questions.
So why you are not tried silver instead
Excellent.
of copper and zinc? Why you are not tried silver nanoparticles? Silver low-grade fibers.
Yeah, thank you so much. This is a very good question.
And so I have second question also. In real-time experiment, you said you are going to perform the real-time experiment. So in real-time experiment, how are you going to apply these nano-loaded natural fibers at the site of injury for wound healing? Is it the dermatological patches or something like that you are going to prepare, sir? How you are going to apply it?
Thank you, madam. Thank you so much for the first question. Why we are not utilizing silver and gold? You didn't ask me gold, but I also want to add gold. Simply because expensive. So we were, of course, copper oxide is not expensive, but it is also more reactive. Huh?
But sir, we will get the synergistic activity because it is proved that it is used for wound healing. So you will get a synergistic activity if you use it.
Yes. Thank you. We could try also do you mean also the combination of both kind of nanoparticles? Yeah. Okay. I understand. You recommend to use silver nanoparticles. Yeah. Yeah. Our intention was to explore the inexpensive variation. Therefore, this is the reason. And the second question was about how are we going to utilize for own healing the nanoparticles for real-time experiment. That is not, we are not going to do that. I was talking about the bone tissue engineering application. Yeah.
But sir, if you want to transfer this technology, then you must suggest us its application, how we can apply it to the wound. Then only this will be really useful, sir. Because you are using a cellulose fiber. We can try several natural fibers also other than cellulose. But how we can apply it to the wound in real time?
So the idea is, I mentioned that this is the work that we are doing. We have not developed so much, but the idea is, I want to tell you, I'm sorry, I hope the picture will come. Yes. This is well known and well established that the silver nanoparticle coated bandage, particularly cotton bandage, when they are moved around the wound, the silver has antibacterial activity and it heals up the wound quickly. And the change of bandage, it takes longer time. Maybe simple cotton bandage you have to change after 3 days or 4 days. But if it is impregnated with silver nanoparticle, it may be changed after 7 days or 8 days. Second question, why copper oxide? Copper oxide is also antibacterial, but it is much cheaper than silver. So economically, it is more economical and more useful. That's why people are trying not only for healing the wound, copper oxides are also being used in construction industry. Titanium dioxide are being used in construction industry for killing the bacteria and other things, especially in the hospitals, and for cleaning the building. Yeah, thank you, sir. Just to add one sentence here. But this is very nice question, madam. Yeah, I was introducing this topic. So we are not working for complex one, for just escaping from bacterial infection or to remove the infection. So if we have a code like this, and if we have a paper like this, which is incorporated, coated with the silver oxide or also silver nanoparticles can be copper oxide or zinc oxide nanoparticles, and that can be put onto this one. And if it helps to avoid the bacterial infection or to eliminate the bacterial infection, that would be a good idea. Yeah, that is the reason. Yeah, thank you. Can you please hold the microphone?
Sir, shall we use bimetallic nanomaterial synthesized from plants, especially copper and magnesium? For those purposes?
I don't know exactly, but in my opinion, yes. Yeah. Yeah. Yeah, thank you. Yeah, as I mentioned, I think yes, but I'm not sure how effective it will be.
Okay, thank you, sir. Thank you very much for providing us the insights about the new biomaterial for the wound healing, especially the bone wound healing. Now, ladies and gentlemen, here we end up with the keynote address. We will shortly be joining here after having a high tea break. There will be the first technical session in which we will be having two invited talks. So please don't forget to assemble here in 10 minutes. Thank you very much.